ये बायोटेक्नोलॉजी एंड इट्स एप्लीकेशन का एनसीईआरटी है जिसमें हम टॉपिक्स देखने वाले हैं बायोटेक्नोलॉजिकल एप्लीकेशन इन एग्रीकल्चर, बायोटेक्नोलॉजिकल एप्लीकेशन इन द मेडिसिन, ट्रांसजेनिक एनिमल्स और एथिकल इश्यूज को। तो एनसीईआरटी ऐसा स्टार्ट करता है कि biotechnology, as you would have learned from the previous chapter, essentially deals with the industrial scale production of the biopharmaceuticals and the biological using the genetically modified microbes, fungi, plants, and the animals. The applications of the biotechnology include the therapeutic, diagnostic, genetically modified crop for the agriculture, processed foods, biodegradation, based treatment, and the energy production. Three critical research areas of the biotechnology are the providing the best catalyst in the form of the improved organism using a microbe or the pure enzyme, creating optimal condition through the engineering for the catalyst to act, and downstream processing technology to purify the protein or organic compound. Or let us now learn how human beings have used biotechnology to improve the quality of the human life, especially in the field of the food production and the health. तो बायोटेक्नोलॉजिकल एप्लीकेशन इन द एग्रीकल्चर में आता है कि लेट अस टू लुक एट द थ्री ऑप्शन दैट कैन बी थॉट फॉर इंक्रीजिंग द फूड प्रोडक्शन। पहला है एग्रोकेमिकल बेस्ड एग्रीकल्चर, दूसरा है ऑर्गेनिक एग्रीकल्चर ऑर्गेनिक एग्रीकल्चर और और तीसरा है जेनेटिकली इंजीनियरिंग क्रॉप बेस्ड एग्रीकल्चर। तो एनसीईआरटी कहता है कि द green revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yields have been partially been due to the use of the improved crop varieties, but mainly due to the use of the better management practices and use of the agrochemicals like the fertilizers and the pesticide. However, for the farmers in the developing world, agrochemicals are often too expensive, and further increases in the yield with existing varieties are not possible using the conventional breeding. As the traditional breeding techniques failed to keep pace with the demand and to provide a sufficiently fast and efficient system for the crop improvement, another technology called the tissue culture got developed. What did the tissue culture mean? It was learned by the scientists in the 1950s that whole plant could be regenerated from the explant. That means any part of a plant taken out and grown in a test tube under sterile conditions in the special nutrient media. This capacity to generate a whole plant from the initial explant is called the totipotency. We will learn to accomplish this in the higher classes. It is important to stress here that the nutrient medium must provide a carbon structure source such as the sucrose and also inorganic salts, vitamins, amino acids, and the growth regulators like the oxygens, cytokinins, etc. By application of this method, it is possible to achieve propagation of a large number of the plants in very short duration. This method of producing the thousands of the plants through the tissue culture is called the micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown. That means they are soma clones. Many important food plants like the tobacco, banana, apple, etc. have been produced on the commercial scale using this method. Try to visit a tissue culture laboratory with your teacher to better understand and appreciate the process. अब आगे है कि another important application of the method is the recovery of the healthy plants from the diseased plants. Even if the plant is infected with a virus, the meristem, apical, and the axillary is free of the virus. Hence, one can remove the meristem and grow it in vitro to obtain the virus-free plants. Scientists have succeeded in culturing meristem of the banana, sugarcane, potato, etc. Scientists have even isolated single cell from the plant, and after digesting their cell balls have been able to isolate naked protoplasts surrounded by the plasma membrane. Isolated protoplasts from the two different varieties of the plant, each having a desirable character, can be fused to get hybrid protoplasts, which can be further grown to form a new plant. These hybrids are called the somatic hybrids, while the process is called the somatic hybridization. Imagine a situation when a protoplast of a tomato is fused with that of the potato, and then they are grown to form a new hybrid plant combining tomato and the potato characteristic. Well, this has been achieved, resulting in the formation of the tomato. Unfortunately, this plant did not have all the desired combination of the characteristics of its commercial utilization. Is there any alternative path that our understanding of genetics can show so that the farmers may obtain maximum yield from their fields? Is there any way to minimize the use of the fertilizers and the chemicals so that their harmful effects on the environment are reduced? Use of the genetically modified crop is made possible solution. Plants, bacteria, fungi, and animal food genes have been altered by the manipulation are called the genetically modified organism (GMO). GM plants have been useful in many ways. Genetically modification has made crops more tolerant to the abiotic stresses, such as cold, drought, fault, heat, reduced reliance on the chemical pesticide, pesticide crop, help to reduce the post-harvest losses, increase efficiency of the mineral usage by the plants. This prevents early exhaustion of the fertility of the soil or enhance nutrition value of the food, example golden rice, vitamin A enriched rice. In addition to this usage, GM has been used to create tailor-made plants to supply alternative resources to industry in the form of the starch, fuels, and the pharmaceuticals. Some of the applications of the biotechnology in agriculture that you will study in detail are the production of the pest-resistant plant, which would decrease in the amount of the pesticide used. Bt toxin is used by a bacterium called the Bacillus thuringiensis Bt for the short. Bt toxin gene has been cloned from the bacteria and had been expressed in the plants to provide resistance to insect without the need for the insecticide. In effect, created a biopesticide. Examples are the Bt cotton, Bt corn, rice, tomato, potato, and soybean, etc. फिर आता है बीटी कॉटन तो कहता है some strains of the Bacillus thuringiensis produce the protein that kill the insects such as the lepidopterans, tobacco budworm, armyworm, phyllopterans, beetles, and dapterans, flies, mosquito. Bacillus thuringiensis form the protein crystal during the particular phase of their growth. These crystals contain a toxic insecticidal protein. Why does this toxin not kill the bacillus? Actually, the Bt toxin protein is used as inactive prototoxin, but once an insect ingests the inactive toxin, it is converted into an active form of the toxin due to the alkaline pH of the gut, which solubilize the crystal. The activated toxin binds to the surface of the midgut epithelial cells and create pores that cause the cell swelling and lysis and eventually cause the death of the insect. फिर कहता है कि a specific Bt toxin genes were isolated from the Bacillus thuringiensis and incorporated into the several crop plants such as the cotton. The choice of the genes depends upon the crop and the targeted pest, as most Bt toxins are insect group specific. The toxin is coded by a gene Cry1F named Cry. These are a number of them. For example, the proteins encoded by the genes Cry1F and the Cry2AB control the cotton bollworm, that of the Cry1AB control the corn borer. तो ऐसे है कि क्राई 1AB जो है 1AB ये हो गया बोरर के लिए बोरर, और 2AB जो होता है ये होता है कॉटन बॉलवर्म के लिए, और उसकी तरफ से 1AC जो होता है ये भी होता है कॉटन बॉलवर्म के लिए। तो इस तरफ है 2AB और 2AC वाला कॉटन बॉलवर्म के लिए। तो यहाँ पर दिए हुए कॉटन बॉल दिया हुआ है, कॉटन बॉल डिस्ट्रॉयड बाय द बॉल बॉम्ब्स, और दूसरा है फुली मैच्योर कॉटन बॉल। फिर आगे है पेस्ट रेसिस्टेंट प्लांट्स। इसमें कहता है कि several nematode parasite a wide variety of plants and animals including the human beings. A nematode Bladogani incognitia affects the root of the tobacco plant and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of the RNA interference. RNA takes place in all eukaryotic organisms as a method of the cellular defense. This method involves silencing of the specific mRNA due to the complementary dfRNA molecule that binds to the and prevent translation of the mRNA silencing. The source of this complementary RNA could be from an infection by the virus having the RNA genome or mobile genetic elements transposons that replicate by an RNA intermediate. Using the agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of the DNA was such that it produced both sense and the antisense RNA in the host cells. These two RNA being complementary to each other formed a double-stranded dfRNA that initiated RNA and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing a specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. तो इधर है जो दिए हुए है कि हाउ, इधर है जो दिखाया हुआ है कि हाउ प्लांट्स generated different triggers protection against the nematode infestation. Roots of the typical control plant, transgenic plant roots 5 days after the deliberate infection of the nematode but protected through the novel mechanism. फिर आता है बायोटेक्नोलॉजिकल एप्लीकेशन इन द मेडिसिन। तो कहता है The recombinant DNA technology process have made immense impact in the area of the healthcare by enabling mass production of the safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological response as in common in case of the similar products isolated from the non-human sources. At present, about 30 recombinant therapeutics have been approved for the human use the world over. In India, 12 of these are presently being marketed. फिर आता है genetically engineered insulin तो कहता है कि management of the adult onset diabetes is possible by taking insulin at the regular time intervals. What would be diabetic person do if enough human insulin were not available? If we discuss this, you would soon realize that one would have to isolate and use insulin from the other animals. Would the insulin isolated from the other animals be as effective as that secreted by the human body itself, and would it not elicit an immune response in the human body? Now imagine if a bacterium were able that could make human insulin, suddenly the whole process becomes so simple. You can easily grow a large quantity of the bacteria and make as much insulin as you need. Think about whether insulin can be orally administered to the diabetic people or not. तो भाई तो नॉट क्योंकि इंसुलिन ये जो प्रोटीन है तो ये जो गट में ही डाइजेस्ट हो जाएगा इसलिए ओरली नहीं दे सकते हैं. फिर आता है कि इंसुलिन यूज फॉर द डायबिटिक but earlier extracted from the pancreas of the slaughtered cattle from the pigs. Insulin from an animal source though caused some patients to develop the allergy or other types of the reaction to the foreign protein. Insulin consists of the two short polypeptide chains, chain A and the chain B, that are linked together by the disulfide bridges. फिर आता है कि in the mammals including humans, insulin is synthesized as the pro-hormone like the pro-enzyme. The pro-hormone alpha is to processed before it becomes a fully mature and the functional hormone which controls an extra upstage called the C-peptide जिसमें 33 अमीनो एसिड होता था. This peptide is not present on the mature insulin and is removed during the maturation into the insulin. The main challenge for the production of the insulin during the rDNA technology by getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of the human insulin and introduced them in the plasmid of the E. coli to produce insulin chain. Chain A and chain B were produced separately अलग अलग plasmid में extracted and combined by creating the disulfide bonds to form the human insulin. अब यहाँ पर दिखा हुआ है मैचुरेशन ऑफ द प्रो इंसुलिन इनटू द इंसुलिन सिंप्लीफाइड करके दिया हुआ है. तो प्रो इंसुलिन है, ए पेप्टाइड है, बी पेप्टाइड है और सी पेप्टाइड को फ्री कर दिए, अलग कर दिए, तब जाके है जो मैच्योर बन गया. आगे जीन थेरेपी है तो इंसर्ट कहता है कि इफ ए पर्सन इज बॉर्न विथ ए हेरिडिटी डिजीज कैन ए करेक्टिव थेरेपी बी टेकन फॉर सच ए डिजीज. जीन थेरेपी इज एन अटेम्प्ट टू डू दिस. Gene therapy is a collection of the method that allow correction of a gene defect that has been diagnosed in a child embryo. Here genes are inserted into a person's cells and tissue to treat a disease. Correction of the genetic defect involves delivery of a normal gene into the individual or the embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy was given in the 1990 to a 4-year-old girl with adenosine deaminase EDA deficiency. This enzyme is crucial for the immune system to function. This disorder is caused due to the deletion of the gene for the adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by the enzyme replacement therapy, in which functional ADA is given to the patient by the injection. But the problem with both of these approaches is that they are not completely curative. As first step towards gene therapy, lymphocytes from the blood of the patient are grown in the culture outside the body. A functional ADA-free DNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires the periodic infusion of such genetically engineered lymphocytes. However, if the gene isolated from the bone marrow cells producing ADA is introduced into the cells at early embryonic stage, it could be a permanent cure. फिर आता है मॉलिकुलर डायग्नोसिस तो कहता है यू नो दैट फॉर द इफेक्टिव ट्रीटमेंट ऑफ ए डिजीज अर्ली डायग्नोसिस अंडरस्टैंडिंग इट्स पैथोफिजियोलॉजी इज वेरी इंपॉर्टेंट यूजिंग कन्वेंशनल मेथड ऑफ डायग्नोसिस सीरम एंड यूरिन एनालिसिस एक्सेट्रा अर्ली डिटेक्शन इज not possible. Recombinant DNA technology, polymerase chain reaction PCR, and the enzyme-linked immunosorbent assay ELISA are some of the techniques that serve the purpose of the early diagnosis. Presence of the pathogen, bacteria, virus, etc. is normally suspected only when the pathogen has produced disease symptoms. By this time, the concentration of the pathogen is already very high in the body. However, very high concentration of the bacteria or the virus at a time when the symptoms of the disease are not yet visible can be detected by the amplification of the nucleic if feed by the PCR. Can you explain how PCR can detect very low amount of the DNA? PCR is now routinely used to detect HIV in the suspected AIDS patient. It is being used to detect a mutation in the gene in the suspected cancer patient too. It is a powerful technique to identify many other genetic disorders. A single-stranded DNA or the RNA tagged with a radioactive molecule probe is allowed to hybridise to its complementary DNA in a clone of the cells, followed by detecting using autoradiography. The clone having the mutated gene will not hence appear on the photographic film because the probe will not have the complementary with the mutated gene. ELISA is based on the principle of the antigen-antibody interaction. Infection by the pathogen can be detected by the presence of the antigens, proteins, glycoprotein, etc., or by detecting the antibodies synthesized against the pathogen. कहता है ट्रांसजेनिक एनिमल्स तो कहता है कि animals that have their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, and the fish have been produced. Although over 95% of all existing transgenic animals are mice. Why are these animals being produced? How can man benefit from such modification? Let us try and explore some of the common reasons. तो पहले नॉर्मल फिजियोलॉजी एंड डेवलपमेंट कहता है कि ट्रांसजेनिक एनिमल्स कैन be especially designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example study of the complex factor involved in the growth such as insulin, like the growth factors by introducing genes from the other species that alter the formation of this factor and studying the biological effects that result information is obtained about the biological role of the factor in the body. फिर आता है स्टडी ऑफ द डे तो कहता है कि मैनी ट्रांसजेनिक एनिमल्स आर डायग्नोस्ड आर डिजाइनेड टू इंक्रीज our understanding of how genes contribute to the development of the disease. These are especially made to serve as models for the human disease so that the investigation of the new treatments for the disease is made possible. Today, transgenic model is used for the many human diseases such as the cancer, cystic fibrosis, rheumatoid arthritis, and the Alzheimer. आगे आता है बायोलॉजिकल प्रोडक्ट्स तो कहता है मेडिसिन रिक्वायर्ड टू ट्रीट द सर्टेन ह्यूमन डिजीज कैन कंटेन द बायोलॉजिकल प्रोडक्ट्स बट सच प्रोडक्ट्स आर ऑफन एक्सपेंसिव टू मेक. Transgenic animals that produce useful biological products can be created by the introduction of the portion of the DNA or genes which codes for a particular product, such as the human protein alpha-1 antitrypsin used to treat the emphysema. Similar attempts are being made for the treatment of the phenylketonuria PKU and the cystic fibrosis. In 1997, the first transgenic cow Roji produced the human protein in Ncert milk, जिसमें 2.4 ग्राम पर लीटर मिल्क प्रोटीन था. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for the human baby than the natural cow milk. आगे वैक्सीन सेफ्टी तो कहता है ट्रांसजेनिक mice are being developed for the use in the testing the safety of the vaccine before they are used on the humans. Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of the monkeys to test the safety of the batches of the vaccine. फिर chemical safety testing तो कहता है this is known as the toxicity safety testing. The procedure is the same as that used for the testing toxicity of the drugs. Transgenic animals are made that carry genes which make them more sensitive to the toxic substance than non-transgenic animals. They are then exposed to the toxic substance and the effect studied. Toxicity testing in such animals will allow us to obtain the results in less time. फिर ethical issues तो कहता है the manipulation of the living organism by the human race cannot go on any further without regulation. Some ethical standards are required to evaluate the mortality of all the human activity that might help or harm the living organism. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of the organism can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organizations such as the GEFC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of the GM research and the safety of the introducing GM organism for the public services. The modification usage of the living organism for the public services as the food and the medicine sources, for example, has also created problems with the patient granted for the same. There is growing public anger that certain companies are being granted patent for the products and technology that make use of the genetic material, plants, and the other biological resources that have long been identified, developed, and used by the farmers and the indigenous people of a specific region या country. फिर कहता है कि rice is an important food grain, the presence of which goes back thousands of years in the Asia agriculture history. There are an estimated 2 lakh varieties of the rice in India alone. The diversity of the rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and the flavor, and 27 documented varieties of the basmati are grown in India. There is reference to the basmati in the ancient text, folklore, and the poetry, as it has been grown for the century. In 1997, an American company got patent right on the basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of the basmati in the US and abroad. This new variety of the basmati had actually been derived from the Indian farmer varieties. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. The patent extends to the functional equivalents, implying that other people selling the basmati rice could be restricted by the patent. Several attempts have also been made to patent usage, products, and the processes based on the Indian traditional herbal medicine, example turmeric, neem. If we were not vigilant and do not immediately counter these patent applications, other countries' individuals may encash on our rich legacy and we may not be able to do anything about it. So, like biopress is a term used to refer to the use of the bioresources by the multinational companies and other organizations without proper authorization from the country and the people concerned without the compensatory payment. Most of the industrialized nations are rich financially but poor in the biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in the biodiversity and the traditional knowledge related to the bioresources. Traditional knowledge related to the bioresources can be exploited to develop modern applications and also can be used to save time, effort, and expenditure during the commercialization. Ncert कहता है कि there has been growing realization of the injustice in adequate compensation and benefit sharing between the developed and the developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of the bioresources and the traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patent Bill that takes such issues into consideration, including the patent terms, emergency provision, and research and development initiative. अब इसका समरी है तो समरी में कहता है कि बायोटेक्नोलॉजी हैज गिवन टू द ह्यूमन सेवरल यूजफुल प्रोडक्ट्स बाय यूजिंग द माइक्रोब्स प्लांट्स एनिमल्स एंड देयर मेटाबॉलिक machinery technique of the tissue culture and the somatic hybridization offer vast potential for the manipulation of the plants in vitro to produce the new varieties. Recombinant DNA technology has made it possible to engineer microbes, plants, and animals such that they have novel capabilities. Genetically modified organisms have been created by using the method other than the natural method to transfer one or more genes from one organism to the another, generally using techniques such as the recombinant DNA technology. GM plants have been useful in increasing the crop yields, reduce post-harvest loss, and make crop more tolerant of the stresses. There are several GM crop plants that, with improved nutritional value of the food and the reduced reliance on the chemical pesticide pest-resistant crop. Recombinant DNA technology poses process have made immense impact in the area of the healthcare by enabling mass production of the safe and more effective therapeutics since the recombinant therapeutics are identical to the human protein. They do not involve unwanted immunological response and are free from the risk of the infection as was observed in the case of the similar product translated from the non-human resources. Human insulin is made up in bacteria, yet its structure is absolutely identical to that of the natural molecule. Transgenic animals are also used to understand how genes contribute to the development of a disease by serving as models for the human disease such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Gene therapy is the insertion of the gene into an individual cell and tissue to treat the disease, especially hereditary disease. It does so by replacing a defective mutant allele with a functional one or gene targeting, which involves gene amplification. Viruses that attack their host and induce their genetic material into the host cell as a part of their replication cycle are used as vectors to transfer healthy genes or more differently portion of the gene. The current interest in the manipulation of the micro plant and animals has raised serious ethical questions. तो ये हो गया बायोटेक्नोलॉजी एप्लीकेशंस का एनसीईआरटी.